Hello children, welcome to biology class. Today we can discuss the topic, the search for genetic material. Now we are aware that the genetic material is DNA or RNA, isn't it? But by 1926, the quest to determine the mechanism for genetic inheritance had reached a molecular level. The question of what molecule was actually the genetic material had not been answered. So, how we could identify the genetic material as nucleic acid? So, first of all, we can discuss the transformation principle. In 1928, Frederick Griffith, in a series of experiments with the bacterium, that is Streptococcus pneumoniae. Children, this bacterium responsible for the disease pneumonia witnessed a miraculous transformation in the bacterium. During the course of his experiment, a living organism, bacteria, that had changed in physical form. So, he identified two strains of bacteria, S strain and R strain. What are the difference between these two strains? This bacterium are grown on a culture plate. Some produce smooth, shiny colonies and he termed it as S strain, while others produce rough colonies. So S means smooth colonies, R stands for rough colonies. Okay. This is because the S strain, which produce smooth colony bacteria, they have a mucus, a polysaccharide coat, while R strain does not. And mice infected with this S strain we can call it as virulent. It is having the presence of that polysaccharide. Which polysaccharide? Mucus coat made up of polysaccharide. They die from pneumonia infection, but mice infected with R strain do not develop pneumonia. Clear? S strain, it is virulent, cause the death of mice, whereas R strain is avirulent. See? So children, you keep it in mind, virulent bacteria, S strain, which cause the death of mouse, whereas R strain, avirulent bacteria, never cause the death of the mouse. Clear? And what was his experiment? Griffith was able to kill bacteria by heating them. Okay? So he observed that heat-killed S strain bacteria injected into mice did not kill them. When he injected a mixture of heat-killed S and live R, yes, of course, the mice died. Moreover, he recovered living S bacterium from the dead mice. See, observe his experiment. R strain, mouse lives. S strain, mouse dies. The first condition. Then, the second condition. What is the second condition? Heat killed S strain. Heat killed S strain. In normal S strain, mouse dies. But in heat killed S strain, yes, mouse lives. Then what is the third condition? Rough strain and heat killed smooth strain. In normal rough strain condition, mouse live, and normal heat killed condition, mouse lives. But see, in this combination of 1 and 3, what happened? Yes, the mouse dies. Children, what may be the reason? Can you identify? Yes, he concluded that the R strain bacteria had somehow been transformed. Okay, so you just notice the sentences in bold. They transformed by the heat killed S strain bacteria. That was the reason. Okay, so some transforming principle, something transformed. Okay, so some transforming principle transferred from heat killed S strain had enabled the R strain to synthesize a smooth polysaccharide coat and it became virulent and it caused the death of the bacteria. And so during the experiment it was clear that some genetic material is transferring, isn't it? And this must be due to the transfer of genetic material. However, the biochemical nature of genetic material was not defined from his experiment. Okay, because the proteins are the while explaining the packaging of nucleic acid, we explain packaging of DNA, we explain the presence of histones. So to identify whether the protein or DNA had transformed as the genetic material, so we can identify one more experiment. This is same experiment, picture of same experiment, that is biochemical characterization of transforming principle by Avery, Colin, then McTree. Okay, Oswald Ivory, Colin MacLeod and MacLean McCarthy. Okay, these three scientists, the genetic material was thought to be a protein. They worked to determine the biochemical nature of transforming principle and Griffith experiment. They purified biochemicals, proteins, DNA and RNA from the heat-killed S cells to see which one could transform live R cell into AS cells and which caused the death of the mice. And after their experiment, they discovered that DNA alone from S bacteria, that means the smooth bacteria, caused R bacteria, rough bacteria, to become transformed. They also discovered that protein digesting enzyme protease and RNA digesting enzymes RNase did not affect transformation. So the transforming substance was not a protein or RNA. Okay, so digestion with DNAs did inhibit transformation, suggesting that the DNA caused the transformation. So they conclude that DNA is the hereditary material. See children, this was their experiment. Here they add proteinase, sample should contain no protein and S up cells appear. Here they add ribonuclease, that means sample should contain no RNA. They add R cells, R cell means rough cells, and S cells appears. But when they add DNase, this is the enzyme, they add R cells and they found no S cells in the sample, and they can conclude that yes, DNA is the genetic material. Clear children? Then now they identified that the genetic material is DNA. And one more proof is there to identify or to conclude DNA as the genetic material, that experiments done by Alfred Hershey and Martha Chase. Okay, we can call this experiment as Hershey-Chase experiment. They worked with viruses that infect bacteria, and it is called, yes, bacteriophage. They grew some viruses in a medium that contained radioactive phosphorus and some others on a medium that contained radioactive sulfur. Which are the two radioactive substances? Yes, radioactive phosphorus and second one, radioactive sulfur. Viruses grown in the presence of radioactive phosphorus contain radioactive DNA but not radioactive protein, because DNA contains phosphorus but protein does not. Similarly, viruses grown on radioactive sulfur contain radioactive protein but not radioactive DNA, because the DNA does not contain sulfur. The same diagram is there in our textbook. See, radioactive phage were allowed to attach E. coli bacteria. Then as infection proceeded, what happened? Yes, the viral coats were removed from the bacteria by agitating them in a blender. The viruses, so that is the first step, infection, then second step, blending, then third step, centrifugation. Then after blending, the viral coats were removed from the bacteria by agitating them in a blender. The virus particles were separated from the bacteria by spinning them in a centrifuge. So, three steps: infection, blending and centrifugation. Bacteria which was infected with viruses that had radioactive DNA were radioactive, indicating that DNA was the genetic material. See, observe this diagram. Here sulfur labeled protein capsule. Here in the colony, what they observed, no sulfur detected in the cell because sulfur was labeled in protein. But sulfur detected in supernatant. At the same time, the phosphorus, radioactive phosphorus were labeled in DNA instead of in protein. And what is the result? Yes, phosphorus detected in the new cells. Clear? That means the genetic material is DNA, not protein. Clear, children? Now let's discuss about RNA. What is expanded form of RNA? Yes, ribonucleic acid. It is the first genetic material. And there is now enough evidences to suggest that essential life processes such as metabolism, translation, splicing evolved around RNA. RNA used to act as a genetic material as well as catalyst. There are some important biochemical reactions in living systems that are catalyzed by RNA catalyst and not by protein enzymes. Children, but RNA being a catalyst was reactive and hence unstable. Therefore, DNA has evolved from RNA with chemical modifications that make it more stable. DNA being double-stranded and having complementary strand further resists changes by evolving a process of repair. So now let's discuss the difference between RNA and DNA. We know that sugar moiety is deoxyribose. Here ribose, adenine, thymine, guanine, cytosine are the nitrogen bases. Here instead of thymine, uracil is present. Then this double-stranded, this single-stranded obeys Charep's rule. They do not obey Charep's rule. Bases are not modified. Here modifications are there. It is stable and not hydrolyzed easily by alkalis. But here it is unable and hydrolyzed easily by alkalis. DNA content is constant in all the cells except during cell division. Here it is varies from cell to cell. The lifespan of DNA is comparatively high. Here RNA is short-lived. No natural DNA is catalytic. Here RNA can be catalytic. Then they present in the nucleus, mitochondria, chloroplast. They present in nucleus, mitochondria, nucleolus, ribosome and cytosol. Okay, so that's about difference between DNA and RNA. Now let's discuss replication. Unwinding, that is replication. While proposing the double helical structure for DNA, Watson and Crick had immediately proposed a scheme for replication of DNA. Okay, and he said that it was not escaped our notice that the specific pairing we have postulated immediately suggests a possible coping mechanism for the genetic material, and suggests that two strands would separate and act as a template for the synthesis of new strand. So after completion of replication, each DNA molecule would have one parental and one newly synthesized strand. It was termed as semi-conservative method. See children, the two strands get separated and two new strands are formed, one continuous and one discontinuous strands, and they are the templates of the parental strand, that is replication. Okay, so by producing a replication fork, the double-stranded DNA get unwind, that is replication, and this process we will discuss in next class in detail. Okay, so today we discussed a lot of things, a lot of experiments. Children, you study it, you read the textbook thoroughly. Okay children, thank you.